Hello and welcome to my channel. If the mechanisms behind breast cancer metastasis is of interest to you, then this is definitely the video for you. I'm going to be going over a paper I've recently published with my old mentor and previous boss, Dr. Paul Sylvester. And this paper is titled Gamma-Tocotrienol Inhibition of Galectin-3 Expression, Distribution, and Oligomerization in Highly Metastatic Breast Cancer Cells. I used mouse and human breast cancer cells in the experiments, and you can find the paper on nature.com or PubMed. And this paper was received in November of 2025 and accepted on the 20th of March 2026. The preprint is out right now, which you can find on the internet, and within a few weeks or months, the final edited version should be available. But you can still look at the preprint right now and see all of the figures I will go through. If you're interested in a deep dive of the figures, please comment because I can make a long technical video on each figure. However, this video is just meant to be a very general overview so that anyone can understand it. So let's continue on to the hypothesis. My hypothesis, to put it short, is that gamma-tocotrienols inhibit the galectin-3 lattice in highly malignant breast cancer cells. Gamma-tocotrienol has been shown to have effects against breast cancer cells, and you can find that in many different papers throughout the literature. And it's been shown to not only inhibit breast cancer cells, but also playing a role in cancer prevention. So there's actually 8 different forms of vitamin E broken down into alpha, beta, gamma, delta tocopherol and alpha, beta, gamma, delta tocotrienol. While they both have shown activity to prevent cancer, since they are an antioxidant, tocotrienols have also shown activity to inhibit breast cancer proliferation. However, the molecular mechanism has been unknown how it's going about doing that. So I looked into the galectin-3 lattice as at least one possible way of how tocotrienols are inhibiting these breast cancer cells. So let's talk a little bit more about galectin-3. Galectin-3 is a partly intrinsically disordered protein that utilizes its intrinsically disordered nature to have multiple conformations, which allows it to bind to itself and form a lattice. And it does that by utilizing that intrinsically disordered portion to mediate liquid-liquid phase separation, which subsequently makes biomolecular condensates, which can combine receptors like the epidermal growth factor receptor to the extracellular matrix. So this is important for metastasis and for proliferation, but it's also important for breast cancer resistance because when this lattice comes into contact with proteins like the MDR1, which is also called P-glycoprotein, which is an ABC transporter, and this ABC transporter is responsible for excreting doxorubicin out of the cell. So that's why it plays a role in breast cancer resistance, because doxorubicin is used as a chemotherapy, the most commonly used chemotherapy against breast cancer. So if the cell can organize those receptors and increase their existence on the membrane, then they will have more of a potential to excrete these chemotherapies. And they can be held on the membrane by these biomolecular condensates. So these biomolecular condensates play a role in metastasis and breast cancer resistance, which is why they are vital targets for new cancer therapies. So again, my hypothesis was that gamma tocotrienols inhibits the galectin-3 lattice, which disengages the breast cancer cells from combining with the extracellular matrix, which will decrease their ability to migrate and metastasize. So first, I started with a Schrodinger molecular docking, and this is just a simulation with gamma tocotrienol and beta lactose. So galectin-3 has a carbohydrate recognition domain, which tocotrienols and beta lactose can bind to. However, gamma tocotrienols can also bind to the hydrophobic recognition domain of galectin-3 with its long hydrophobic tail. So while gamma tocotrienol and beta lactose both bind to the carbohydrate recognition domain, and similarities in binding are with arginine and glutamic acid, which can be shown right here on beta lactose and right here with tocotrienol. Tocotrienol can also bind the hydrophobic binding region, which is important in its oligomerization. Both of these binding sites are important for its oligomerization. The only thing missing here, I think that might play a role, is the intrinsically disordered tail probably does get inhibited too by gamma tocotrienol. However, this specific docking assay doesn't really look into that. So I didn't have the molecular software to look into the dynamics of the intrinsically disordered tail coming in contact with the binding pockets on galectin-3. However, these simulated results did show that gamma tocotrienol does have similar binding to beta lactose, which beta lactose is used in many laboratory experiments as a competitive galectin inhibitor. So these are just simulated results to give us an idea of what might be going on with the molecular binding. However, to really see what's going on in real cells, we need to look at the proteins, the protein structure, and the protein expression. So to evaluate the protein expression, we use Western blot semi-quantitative analysis, which uses the total protein in the gel to normalize the blots that you get. So you can see that over a 12-hour period, the galectin-3 expression slowly went down and decreased. And the way you can make this a statistical analysis is by using the total protein to normalize it. So you can see that the total protein stayed the same while the galectin-3 expression in the blots decrease, which shows a decrease in galectin-3 expression during a 5 micromolar gamma tocotrienol treatment. So that gives us an idea that gamma tocotrienols do decrease the galectin-3 protein expression in breast cancer cells. So to look at the distribution of galectin-3 in these breast cancer cells, we utilize immunocytochemistry, which uses fluorescently tagged antibodies to bind to galectin-3 so we can see where it is. And you can either utilize Triton X100 or you cannot utilize it. And what Triton X does is it punctures little holes in the membrane. You just want to use 0.4% so that it just makes little holes in the membrane and it doesn't wash it all away. But that allows you to visualize proteins inside the cell, since the antibodies can go through those holes in the membrane, go through the holes in the nuclear membrane, and visualize galectin-3 in the nucleus, as you can see here. And whenever you don't use Triton X100, then you can visualize the galectin-3 expression outside the cell. And this is consistent with previous results showing that in highly metastatic breast cancer cells, galectin-3 is expressed in the nucleus and outside the cell. However, I was interested in looking at the galectin-3 activity outside of the cell, because that's mainly where the interactions happen with the extracellular matrix. So for the rest of the experiments, I mainly didn't use Triton-X unless I was looking inside of the cell. So to look a little closer at the morphology of galectin-3 outside the cell, I used a higher magnification than in the previous immunocytochemistry images I showed you, and you can see the bubble-like structure, or maybe droplet-like structure of galectin-3 biomolecular condensates, and this was outside the cell. You can see it in green right here, and with 5 micromolar of gamma tocotrienol, that gets disrupted, and those structures aren't present anymore. And you can also see a structural analysis in black and white, and scientists do this just so you can have better contrast and clearly see the structure. Moving on, I wanted to look at the fibril formation. So fibronectin in the extracellular matrix forms little fibers, little microscopic fibers that are organized by galectin-3. This has been shown in previous studies. And in the control cells, you can clearly see this occurring. And then whenever you treat the cells with tocotrienol and beta-lactose, you see an absence of these fibrils. So these fibrils also align with the intracellular F-actin cytoskeleton, and what these protein complex make are these lamopodial protrusions that you can see here, which are important for the cell migration and metastasis. And when you treat the cells with gamma tocotrienol or beta lactose, you see that these elongated protrusions get disrupted. So next we wanted to see the exact protein-protein organization and protein-protein binding of galectin-3. So we use a FRET assay, which is a fluorescence resonance energy transfer. Another thing it's called is Foster resonance energy transfer. And what this assay utilizes is 2 different fluorophores bound to galectin-3, and one of them can be excited with 488 nanometer wavelength to produce green fluorescence, and then the other fluorophore can be excited with 543 nanometers to produce red fluorescence. However, when both of these fluorophores are within close proximity to each other, within 10 nanometers, then the donor and the acceptor fluorophore can transfer the energy so that the 488 nanometer fluorophore can actually excite the red fluorophore to produce red light. So you can see normally the 488 nanometer fluorophore makes green light, but when it's in close enough proximity with the 543 nanometer fluorophore, then the energy gets transferred and red light is produced. So this is a way to see that these fluorophores that are attached to the galectin-3 proteins are within close proximity, so that you can see and measure the oligomerization of the galectin-3 protein. So under control conditions, you could see a FRET signal, and then under the treatment conditions, the FRET signal does not appear, which suggests that galectin-3 oligomerization is not occurring. So we wanted to do this in living MDA and B231 cells, which are highly malignant human breast cancer cells. So we used the microplate reader to get the FRET signal. And again, these are in living cells, where the previous image was on fixed cells using microscopy. So you can see that after a 1 and 6 hour treatment period, you had a decrease in FRET signal whenever tocotrienol and beta lactose was used. So next we wanted to use immunocytochemistry so we can investigate how these galectin-3 inhibitors could be used with current treatments, for instance, doxorubicin, which I talked about earlier as being one of the chemotherapies that are commonly used in breast cancer treatment, which the cells can form resistance to and pump out of the cell by a protein called P-glycoprotein, or another name for it is the MDR1 ABC transporter. And doxorubicin, you can see it in red because it's very useful for experimentation because it emits a red light. So whenever you use it, you don't have to use antibodies or anything. You can just measure where it is by the light that it's emitting. So you can see in the control cells, we didn't use any doxorubicin, it's not there. And then in 2 micromolar of doxorubicin, it is present inside the nucleus where its main activity happens, where it breaks the DNA. And then whenever tocotrienol or beta-lactose was used, you can see an increase in doxorubicin in the nucleus. And then you can also not only see it in red right here, but you can see the colocalization of doxorubicin with the DAPI nuclear counterstain, which was blue. So when the blue and the red mix, you get this magenta color that you can see right here in the tocotrienol and beta-lactose. And whenever I copied this onto the presentation, the beta-lactose got cut off right here. But if you look at the preprint on the internet, it will be shown. And this is just giving an example of how tocotrienols could be used with current treatments, but also tocotrienols are found in many diets, dietary supplementation. So it's also an example of how dietary supplementation can help current therapies work even better. And that this is already being used in some of the more advanced research hospitals. However, this does give a mechanism of how these diets are potentiating these treatments. So for conclusion, this paper was just in a laboratory setting, uh, in a dish on cells. So this is just a mechanistic paper. More research will have to be done in animals to really see what's going on and evaluate how this can be used in humans. However, I do think this shows great potential for not only targeting galectin-3, but biomolecular condensates in general outside of the cell, because their dynamics can mediate cancer cell resistance and metastasis. So gamma tocotrienol influence galectin-3 biology in aggressive breast cancer cells. Galectin-3 plays an important role in cancer behavior. Cancer depends on molecular organization, not just mutations, and that's the organization of the extracellular matrix and receptors to create these adhesions that mediate metastasis, that organize via liquid-liquid phase separation into biomolecular condensates. And these findings improve our understanding of cancer biology. And as I said before, this is just a laboratory study, so it does have limitations. More research is needed. Additional studies, including animal models and clinical trials, are necessary to determine how it can be used in humans. And the broader scientific message is understanding how proteins like galectin-3 organize and function inside cells may help scientists identify new strategies to target cancer and disrupt the systems that metastatic cells rely on, like biomolecular condensates, as I've mentioned a few times. So I hope you enjoyed this video. It gives you a look at what goes on in molecular biology labs. And please comment with any questions, or if you want to dive deeper in the results, or maybe just how the experiments work, please comment and I can make more videos. Hope you have a good one.